This
PAHs,
conference will now be recorded.
mine leachate, and emerging contaminants. Appropriately enough, he has bachelor's and master's of science degrees in geology from the University of Texas at Austin. And since 2014, Austin has been involved with numerous freshwater microplastic studies focused on sources, occurrence, transport, fate, and biological uptake. So with that, I will transfer it over to Austin.
Sorry, let me just interject for those who may have heard a message. I am recording this, and we'll send out the link to that afterwards.
Okay, thanks for the introduction, Mark. So I'm going to be talking about, like the title suggests, microplastics in Lake Mead National Recreation Area. So this is a study between USGS and Park Service that we just published earlier this year. We did the field work for a couple of years ago. Here we go. So I'll spend some time talking about background on microplastics for those of you who may be new to the topic. We'll talk about what microplastics are, different sources, different types, pathways to the aquatic environment, a little bit about biological effects, and then I'll focus most of the talk on results of this study on occurrence and uptake in Lake Mead National Recreation Area. And then I'm going to spend a few slides at the end talking about atmospheric deposition of microplastics, which is one of the kind of the cutting edge fields of microplastics right now that I find especially interesting. Okay, so first of all, what are microplastics? Microplastics are any plastic particle sized from about 100 nanometers up to 5 millimeters. So it covers a large range of sizes of plastic particles and a lot of different types of particles. Smaller than microplastics are nanoplastics, which we know exist, and we know that they're pretty abundant in the environment, but we actually have a pretty hard time measuring nanoplastics because of their difficulty and the current lab methods are hard to do that. And actually, a lot of microplastics are pretty hard to measure. Most labs currently can only measure reliably down to 100 microns, or maybe 10 or 20 microns for some of the best labs. Less than 10 or 100 microns, it just gets really difficult to tell if a particle is plastic or if it's some kind of natural fiber or natural material. So we're going to be focusing on this little window of microplastics right here, 100 microns to 5 millimeters. Okay, so we generally talk about microplastics types in terms of their shape. So different shapes or types of microplastics include lines, things like pieces from nets or ropes or fishing line, beads or pellets, or anything spherical. These might come from personal care products like exfoliating face scrubs, that kind of thing. Also pre-production pellets, which are used to make other plastic items. Foams, things like styrofoam, coffee cups, takeout containers, packing peanut type things. Films from things like plastic bags and wrappers. Fragments is kind of a catch-all category of just broken up larger things. Fibers from things like clothing and textiles and carpet and upholstery. And then lastly, tire particles from actual tire wear on the road, but also from chrome rubber, which is used in sports fields, shown here, and even rubberized asphalt, which I only learned about fairly recently. But this is asphalt that they actually grind up old used tires and mix it into the asphalt, which is a great way to get rid of old tires, but maybe not a great thing for the nearby stream ecosystem. So what risks do microplastics pose to aquatic organisms? organisms are definitely ingesting plastics. This has been observed in well over 100 species, probably several hundred species now. Everything from marine mammals to birds, fish, turtles, invertebrates, etc. Pretty much everywhere we look, plastics are being ingested. But the real question, and this is one of the big questions in the field right now, is what risk do they pose? So the potential risks are both mechanical and chemical. So by mechanical risks, I'm talking about plastic particles clogging feeding appendages or creating a false sense of satiation, meaning that the organism thinks it has a full belly, but it's really just a bunch of plastic in its belly, which obviously doesn't deliver nutrients. Stomach ulceration, things like that. So those are mechanical risks or potential mechanical risks. On the chemical side, there are a lot of different chemicals that are added to plastic in the manufacturing process, things like flame retardants, alkyl phenols, antimicrobials, plasticizers, styrenes, antioxidants, etc. These things are manufactured into the plastic to give the plastic different properties. But the concern is that if a fish or other organism eats the plastic, that these chemicals can then leach out into the organism. In addition to what's manufactured in the plastic is chemicals that sorb to the surface of the plastic when the plastic is floating around in the water or out in the environment. For anybody that's used passive samplers like SPMDs or POSIS knows that plastic is really good at sorbing contaminants. Things like PCBs, DDT, PAHs, also metals and pathogens. A lot of these chemicals don't like to be floating freely in the water. They like to attach to either float sediment or plastic floating by. So these chemicals again sorb to the plastic, and then the concern again is that they might be transferred to the organism if the plastic is eaten. So this slide is a little bit out of date. There have been a lot of lab studies on effects of ingestion of plastics, and a lot of those, I'd say the majority of them do document negative effects. Some of them do not. I think it really comes down to the size of the plastic, the shape of the plastic, the composition of the plastic, whether it's polystyrene or polyethylene or polypropylene, etc. What chemicals are the additives in the plastic. There's a lot of different variables to this. So it's hard to say, it's hard to set a standard because of all these different variables. And in the natural environment, the effects are even less clear because there's so many compounding variables that it's hard to isolate the effect of plastic versus the effect of other stressors. Pathways to the environment include wastewater treatment plant discharges, but wastewater treatment plants are actually quite good at removing plastics from water. Obviously, they're not designed to do that, but depending on the level of treatment, they can remove 95 to 99% of the plastics coming in. A lot of that ends up in their biosolids, though, and about 50% of the time, those biosolids are then applied over land. So that's another potential source, or is a source to the aquatic environment, is these biosolids applied over fields, and then those particles washing into the nearby stream or being picked up by the wind and transported away. Stormwater runoff is another pathway, and then atmospheric deposition, which we'll talk about again at the end. All right, so that's it for background information. So now we're going to move on to talking about some results from this study of microplastics in Lake Mead National Recreation Area. So this is a study with the Park Service. The main questions behind this study were, are microplastics present, and do concentrations of microplastics reflect human impacts, or areas of the lake that receive more human use, do we see more microplastics in those areas? And then the second question being, are microplastics being ingested by aquatic organisms? So we sampled 9 locations shown here on the map. We sampled water at all the locations and sediment at all the locations. We also sampled fish and bivalves at a subset of the locations. And then we also did some sediment cores in Las Vegas Bay to try to look at concentrations over time. I'm not going to present those results today just because of time. So the way we sample microplastics typically is using, so this is the way we sample in water, using a net like the one shown here. So we'll drag this net in the surface of the water. So we're sampling just that surface layer. This net is about 70 centimeters across by 40 centimeters tall. So we're sampling like the upper 20 centimeters with this net. The mesh on this is 100 microns. We tow it for about 15 minutes. These floats keep it at a constant depth, and this flow meter in the mouth, you can see right there, we use to then calculate the volume of water that's passed through the net so that we can calculate the concentration of plastics. After towing it, we hang the net up and spray it down with a high pressure hose to get all the material down to the very bottom of the net, which is shown here, and then we can transfer all that material into glass jars for analysis at the lab. Sampling sediment, we use like a dredge, like the one shown here, like a Ponar or a Ekman type dredge, which we just lower down and grab some sediment and bring it back up. This picture shows our winch broke, so we had to pull these things up by hand, which if you've been to Lake Mead, it's pretty deep. So this was a real chore getting some of these back up. But then once it was up, we just were interested in the uppermost sediment, so we sampled just the top few centimeters. Bivalves, we targeted quagga mussels for our bivalves because they're abundant and everywhere in Lake Mead. We wanted to see if we saw differences in concentrations in a heavily impacted area, which is Las Vegas Bay and the boat harbor shown here, versus a less impacted area, which is Overton Arm shown up in the north. So we sampled quagga mussels at both locations. Because quagga mussels are so small, we composited 10 to 11 individuals into a single sample for each site. We also opportunistically caught some Asian clams in some of our sediment samples, so we analyzed those as well. We only had about 3 of those. And then fish, we had the same approach. We collected fish from an impacted site, Las Vegas Bay, and then a less impacted site, Overton Arm. And then we also wanted to look at pelagic versus benthic feeders, so we targeted striped bass and carp. All right, so here are the results. In water, these are the water concentrations. So here are the concentrations in map view. So concentrations range from less than 1 particle per cubic meter of water to up to around 10 particles per cubic meter of water. The highest concentrations were in Las Vegas Wash, which is what we expected because Las Vegas Wash is coming out of Las Vegas, and it's actually mostly effluent from Las Vegas. So we did see the highest concentrations there, as expected. This plot on the left shows the plastic types in each of these samples. So you can see that for pretty much every site, what we saw the most was plastic fibers. To compare these concentrations to a previous study in Great Lakes tributaries, the plot on the right now is 29 tributaries to the Great Lakes across 6 different states. Just to give you a comparison, so Las Vegas wash coming in around 10 particles per cubic meter is, it's comparable to some of the higher concentrations we saw in Great Lakes tributaries. But the rest of the sites in Lake Mead were pretty low, less than 1 particle per cubic meter to 2, maybe 3. So these are pretty low concentrations, and really most of these are right around the detection level. So that's good news for Lake Mead. This comparison is not quite a fair comparison because these are all rivers, and most of these sites are lake sites. And in a lake, a lot of particles will sink. Once they reach the lake, because of their density, they'll sink. So that's why we also sample sediment. So here are concentrations in sediment. Sediment concentrations range from a few hundred particles per kilogram dry weight up to 1,000 particles per kilogram dry weight. Interestingly, Las Vegas wash, where we saw our highest water concentration, was our lowest sediment concentration. And I think that's because this was a pretty quickly flowing stream, and so those particles just don't have a chance to deposit in Las Vegas wash. The water's moving too fast for them to deposit. Again, we see mostly fibers in the sediment samples, and we also saw at the boat harbor, we saw tire particles. If you've ever been to the boat harbor in Lake Mead, it's surrounded by this giant wall of tires shown here. So maybe not a surprise that we see some tire particles in the sample from the boat harbor. To compare the Lake Mead sediment samples to a couple other locations, so this middle plot is the Delaware National Scenic River, and then the plot on the right is sites around Milwaukee, some streams and some harbor sites in Milwaukee. The y-axis for Lake Mead and the Delaware are the same, but the y-axis on the Milwaukee plot is different, so be aware of that. So the concentrations in Lake Mead are comparable to what we saw in the Delaware River, but much lower than some of the sites we saw in Milwaukee that were getting up into the 2,000 and 6,000 particles per kilogram range, and a lot of tire particles in some of those sites. All right, bivalves. Like I said, we were targeting mainly quagga mussels at 2 different sites, a site that was heavily impacted, Las Vegas Bay, and then a site that had minimal human impact, which is Overton Arm. And again, these are averages of 10 to 11 different individuals for these quagga mussel samples. So we did see a pretty big difference between these 2 sites. The site with the more impact, we saw about 12 or 13 particles per organism versus about 2.5. 13 or 14 particles per quagga mussel is quite a lot given the size of a quagga mussel. These things are about the size of like your thumb fingernail. So that's a lot of plastic to pack into a little tiny organism. The few Asian clams that we just happened to collect, we analyzed. Those are all from pretty impacted areas, the harbor, the boat harbor, and Las Vegas Bay. And these things are a little bit bigger than quaggas. They're more the size of like a quarter or a silver dollar. But these things had really high concentrations of plastics, up to over 100 plastics in a single clam. So that was a pretty surprising concentration. Like the water and the sediment samples, these are mostly plastic fibers that we're seeing in the bivalves. And then lastly, in the fish, again, mostly we see fibers ranging from no particles up to about 18, 19 particles per organism. If we compare our locations, Las Vegas wash bass versus Overton arm bass, we definitely saw higher concentrations in Las Vegas wash, Las Vegas Bay area. And then if we compare Las Vegas wash carp to Las Vegas wash bass, the benthic feeder to the pelagic feeder, we seem to see higher concentrations in that benthic feeding carp than we do in the bass. So I think we've already talked about it, but just to review that, we see fibers as the dominant particle type in all of our sample types: water, sediment, bivalves, and fish, all overwhelmingly fibers. So this, that we see fibers pretty much everywhere we sample, even the pretty remote areas that we've sampled, we seem to see these fibers. That has raised questions about how these fibers are getting to these remote areas. So now we're going to shift to atmospheric deposition as a pathway to remote environments. So we had this question of is the atmosphere potentially delivering plastic fibers to remote environments? So we thought a way to test that would be like a Alpine Glacier environment where the only plastics might be coming from the atmosphere. This is not a place where humans are recreating much, if at all. And so if we see plastics up there, then it has to be coming from atmospheric deposition. So we did a pilot study. We only have a handful of samples so far in North Cascades National Park. So here's a map of Seattle, Vancouver, Tacoma area, and North Cascades is just east of Vancouver and Seattle. So we worked with the Park Service to collect a few snow cores from the glacier on a couple different glaciers in North Cascades to look at microplastic concentration in those cores. So they collected cores down to 4 to 6 meters depth, which represents less than a year of snow depth. So it's not a core through time. It's just the most recent year. And then they melted that core down, and we analyzed the melt for microplastics. So here are those results, and notice that the axis here is particles per liter, not particles per cubic meter like we were reporting for Lake Mead. So about 1,000 times more concentrated here. So concentrations in these glacier samples were 10 to 30 particles per liter of water, which is about 1,000 times higher than what we were seeing in Lake Mead and what we see commonly in rivers. So these concentrations are comparable to what we see in urban runoff, which is just staggering that we're seeing such high concentrations in these remote alpine environments. Around the same time, we were working on a study in Denver and Rocky Mountain National Park. This was led by Greg Weatherby, the USGS. Greg is involved in the National Atmospheric Deposition Program. So this is one of their samplers. They collect both wet and dry samples. So the way these work is they have a sensor, and when it starts raining, this lid will flip over to the other bucket, so it will collect a wet sample, wet deposition sample, and then when it stops raining, it flips back, and so then they're collecting a dry deposition sample separately. So they have a network of sites in the Denver, well, all over the US, but Greg was focusing on Denver and Rocky Mountain National Park. And he wasn't actually sampling for microplastics. This wasn't a study designed for microplastics. He was looking at nutrients in rainfall samples. But when he filtered those rain samples and looked at them under the microscope, he started seeing all these little plastic particles. Most of them were plastic fibers. Some of them were more like chunks of plastic. But he found plastics in over 90% of his rain samples. He saw more at the more urban sites down in Denver, but he also saw plastics in samples from Loch Vale up at Rocky Mountain National Park at an elevation of 10,000 feet. He even saw tire particles in some of his samples. This is a scanning electron microscope image of what we're pretty sure is a tire particle. And this was from a rain sample at Loch Vale, again at 10,000 feet in Rocky Mountain National Park. So Janice Brainey, who's a researcher at University of Utah or Utah State, I can't remember, but she used these NADP samples and took it a step further. And you may have seen this just came out in Science this last week. So she looked at these NADP samples from wet and dry deposition samplers at about a dozen places around the western US. These are all protected. These are all in national parks and protected areas. And she found deposition rates averaged 132 pieces of plastic per square meter per day in these locations, which she extrapolated to equal over 1,000 metric tons of plastic deposition to the western US protected lands annually. So to conclude, microplastics and especially microfibers are not limited to urban areas, but appear to be ubiquitous in aquatic environments and are being ingested by aquatic organisms. The risk of ingestion is still somewhat poorly understood. And atmospheric deposition appears to be an important pathway to aquatic environments, and in remote areas, atmospheric deposition is probably the primary pathway. But we do need more research on that topic, because this is like I said, this is kind of the cutting edge topic in microplastics research. And just some of the questions that we still have on this are like, are the microplastics coming from the local nearby urban area, or are they, is this long range transport from Asia or somewhere else? So with that, I'd just like to thank some folks at USGS and the Park Service and funding from the USGS National Park Service Water Quality Partnership. And I think we have plenty of time for questions if there are questions. So thanks everybody.
Great. Thanks, Austin. That was amazing. Those are big numbers, especially that last one about falling on protected areas and all. So I see some chat items. Mark can sort of prompt those, and if there's any follow-up from the people who wrote them, you're welcome to turn yourself off mute to add something to it.
Cool. So first question we got is from Shauna Witt, and it's, what methods do you use to separate the different types of particles from one another when they're extremely small?
Yeah, so I don't analyze the samples myself. I have a colleague in Tacoma who basically operates the lab for USGS. They go through a series of steps to first digest all the organic material in the sample, like the leaves and sticks and algae, and then some sieving steps, and then filtering it, and then it's basically just picking through the sample under a microscope to identify and count and classify the particles. So they just use some tweezers and a microscope essentially to pick through them.
Cool. Next question's from Dave. It's what time of year was the sampling? Was there thermal stratification of the lake occurring?
Yeah, that's a good question. I didn't include it. So that we sampled in March, and I'm gonna zip back real quick because it's kind of interesting. So in our water samples, we had fairly low concentrations over here in Greg Basin, which is where the Colorado River comes in. So that at the water surface, concentrations were relatively low at Greg Basin at the mouth of the Colorado. But then in our sediment samples, the same location, we had our highest concentration of sediment in sediment samples, which was, was kind of puzzling why we'd have such high concentrations in sediment and such low concentrations in water. And I think the answer is that Colorado River water coming in is often colder, and so it's kind of diving beneath, plunging beneath the lake surface, and so it's it's depositing its load of microplastics, but you're not necessarily seeing that at the surface. The surface is lake water, and down deep is basically river water. So I guess that kind of answers the question. As far as stratification, I'm not sure whether it was stratified or not, but from what I'm told, this plunging of the Colorado River water, it would be expected at that time of year.
Next one's from G. Paulson. What's the average size of particles associated with atmospheric deposition?
So that, like the particles that Janice Brainy was analyzing were in the 20 microns up to 5 millimeter range. And I think she had a pretty, I don't remember her numbers exactly, but I think it was a pretty similar split between particles of like 20 to 100 microns and then 100 microns to like 5 millimeters. But especially those fibers, because of their shape, they can be picked up and carried pretty easily. Even a somewhat large fiber can be carried by the wind pretty easily.
This one's from Paolo Lombardo. Have you looked at, or are you planning to look at microbiota attached to plastics entering water bodies in future studies?
Microbiota attached to plastics? No, we haven't. I haven't done that, but others have. They've looked at colonization of plastics, for example, upstream and downstream of wastewater treatment plants, and they've seen different colonization patterns downstream of a wastewater treatment plant than upstream, and different pathogens attached to the plastics upstream versus downstream.
Next one's from Mark. How do you sanitize sampling equipment in sample jars to minimize contamination?
Well, we don't necessarily need to sanitize, but contamination is a really big issue. Actually, So we often just use like a glass mason type jar. And what we've sent some of those jars to the lab, unopened, just out of the box, and send them to the lab, and they often have a few pieces of plastic in them, just probably from the air in the factory. So it is important to rinse the jars and the sampling equipment well before you use it. And during the sampling process and during the analysis process, contamination from the atmosphere, from what you're wearing, and just from the atmosphere is a big concern and something you really need to be thinking about.
I just saw Paola posted a response. She says, thanks, I was thinking about microbiota transported across habitats with plastics.
Oh, interesting.
Microbiota.
Yeah, I don't, that would especially be interesting, if you're talking about wind transport. I think, transporting from different habitats across potentially large distances, that's an interesting thought.
And then I had a question. Are fibers the most pervasive in the environment because they're the most commonly produced types of plastic, or is it because they're transported the most easily, or maybe because they're ingested by a biota preferentially?
I think it's because they're so the inputs are highest, because we fibers from clothing, the amount of fibers that wash off of your clothing both during washing and during drying, it's kind of staggering. It's like thousands of pieces per article of clothing per washing and drying. So I think it's because there are just more of those entering the environment.
Do you think any of the efforts from companies to reduce microfiber use, like Patagonia, have had any effect or will have any effect on deposition or ingestion?
I know that Patagonia and potentially other companies are interested in this. I don't know how far they've gotten in terms of coming up with newly engineered fabrics. I haven't seen them advertising anything like that. So I guess I would say I don't know. I don't know that there are any products out there with that goal yet.
Thanks. Next one's from Dave again. If there's atmospheric deposition of microplastics less than 100 microns in diameter, could the lake samples with the 100 micron plankton net miss much smaller particles? And if so, do you have any estimates on how much?
Oh yeah, definitely. I should have mentioned that. So there have been some studies that have looked at, they basically look at large particles and then down to the tens of micron size particles, and they see exponentially more of those particles in the tens of microns range than in the hundreds of microns range or the millimeter range. So yes, we're definitely missing particles with our 100 micron net, and probably exponentially more than what we sample. So what we sample and what the results I've shown you are probably just as the tip of the iceberg in terms of the numbers of plastic pieces out there.
All right, well, it looks like we got through all the questions here. So unless I see one in the next couple seconds or so, yeah, I just want to thank you a lot, Austin. That was a great presentation. And I'm going to be posting a link in the chat right now. This is to the CALMS post. Wait, I think I posted the wrong one. My bad. I'm going to post a link in a second for our post-webinar survey. And yeah, I'd appreciate it if you guys would take that so we can keep running these and keep everyone happy. Here it is right now. Cool. All right. Well, yeah, thanks a lot. And thanks everyone for joining.
Thanks, everybody.
Thanks, Boston. Take care, everybody.